Hello and welcome to the talk Bee Health Monitoring in Austria. It is about the surveillance study we have done 5 years ago during the project Future of the Honeybee, in German Zukunft Biene. My name is Dr. Linde Morawetz. I'm working for the Austrian Agency for Health and Food Safety in the Department for Apiculture and Bee Protection. The surveillance study we have done in Austria was part of the National Bee Health Project Future of the Honeybee. It was done in the year of 2014-2017. The surveillance study itself was conducted between summer 2015 and spring 2016. It involved 200 apiaries all over Austria. The bee inspectors inspected in that time around 2,000 colonies. They did 3 inspections, one in summer, one in autumn and one in spring. You see, we chose the times of inspection in a way that we saw the condition of the colonies before winter, that means in summer and autumn, and after winter to see which colonies survived. The research questions of the study were the clinical prevalence of bee pests and diseases, data I will not show you today, and the reasons for winter losses in Austria. The data collection was done by professional bee inspectors, which we also trained for the project. When they visited the beekeepers, they began with an interview. They asked them about their beekeeping education, about their operation size, about the type of the operation and so on. They also asked very specific about varroa treatment. Afterwards, they began to inspect the colonies. When there were 10 colonies or less, they inspected all colonies. When there were more, they inspected it after a certain key. So they noted the colony characteristics such as the age of the queen, the size of the colony, or problems with the colony. When they found colonies, you see problems, as you see on the photos, for example, bees with deformed wings or a patchy brood pattern, they took samples with these symptoms and sent them into our lab for analysis of the disease. They also sampled for bees and bee bread in all of the colonies. The bees were sampled to get the varroa infestation rate on bees. We did this in the lab, and then they were sampling for another study, the postdoc study. In the end, we had 27 factors which we could correlate or test with winter losses. These were factors of the beekeeper characteristic, such as number of colonies or experience of the beekeeper. It was factors of beekeeping practice, such as queen age, type of winter food, and so on. There were also unspecific signs of colony problems. For example, this could be the colony size, a very small colony size, or queen failure. We also had the factor parasites, in specificity the varroa mites per adult bees in the model, and also disease during summer and autumn inspection. This could be American foulbrood, varrosis, nosemosis, and so on. With all these 27 factors, we calculated a general linear mix model with binomial distribution. I don't go into detail here, but in the end, this model selected 4 factors which were correlated with winter losses in Austria. The first and most important factor was the varroa infestation level, and it showed that the probability of winter losses could be up to 100% winter loss. So the model gave us a probability that a colony may survive or die. And in varroa, this probability to die could be 100%. So when you look at the graph, you have the x-axis where you see the percentage varroa mites per bees in autumn, and the y-axis you see the predicted probability. And you see the curve increases both positively. So a colony with an infestation level of 10% mites on bees had a loss probability of 10%. A colony, for example, with a very high mite infestation rate of 40% had a loss probability of 80%. So there was a very high probability of loss. These results confirm studies of our results of other European countries, so such in Germany or the Netherlands, or also the Pan-Europe project Epiloby. The second factor, we found the significance with correlation with winter loss was the experience of the beekeepers. Shortly said, the more experience a beekeeper had, the less winter losses he experienced. Again, you see in the graph the probability, but here you see the scale is much lower, so the highest probability was 12% here, and you see the years of experience. It takes nearly 20 years of experience to reduce the probability of winter loss by 50%, so this factor is an important factor, but it takes a long time to change this factor. The years of experience may be a measurement for professionality, so the more experience a beekeeper has, the more professional he or she acts. EpiLobby, the pan-European project, showed also a correlation between professionality and loss. So large beekeeping operations, which are more professional beekeeping operations, showed lower losses. On the other hand, EpiLobby showed that old beekeepers had also high losses, which means it's a little bit contradictory to our results. But maybe there's a point where a beekeeper is not experienced anymore, but too old to be able to adapt to changes, to new techniques, and so he or she gets problems with the time. The third factors we found was colony size in autumn. The graph shows that weak colonies had a much higher probability to die than strong or normal colonies. The colony size is the size of the colony during autumn inspection. But you also see again the scale, which is only up to 12% probability. Again, it's not a very strong factor. This weak colony is a very unspecific symptom for diseases or other problems, and here we can ask, is an important factor lacking, which is the reason for this disease or this symptom? In the German bee monitoring DBMO, it was also showed the small colonies are a predictor for losses. The fourth factor was the age of the queen. It showed that a queen which was 2 years old or more had produced higher losses than younger queens. It was a risk multiplication with a factor of 3. Old queens are said to have a reduced worker production, so they don't lay that many eggs, and old queens are correlated with short-lived winter bees, which means that maybe an old queen is not able to produce bees which have the lifespan to live until spring. Again, the German bee monitoring DBMO showed also that queen age is a predictor for loss. So our results are comparable to the results of the neighbor country, Germany. After the results, I now have to show you that the year we did our study in was a very special year. You here see the colony loss rate in winter in Austria. It's from the colors monitoring of Robert Brotschneider. And you see that the year we did the study in 2015 and 16 was the year with the fewest losses till this monitoring began. So we'd had a year with few problems, and that may be also found in the model. So when we in the end ask what did the model tell us or what didn't the model tell us, we have to say it selected the most relevant factors for Austria for winter loss, but it only described 1 year of sampling, which means that this special year may produce data which another year wouldn't have. So in a year with high losses, there may be other factors, different factors, or also other scale ranges than I showed you here. And it may be that some factors may need a higher sample size. We sampled 200 colonies, which is a lot for such a small country and such a high effort, but with other methods you could have a higher sample size, for example, only interviewing and find other factors which need a higher sample size. It ranked the significant factors, the models. So I showed you that the varroa mite had the highest impact in the model. The losses were up to 100%. With the other 3 factors, the loss probability was only up to 10%. And we also have to speak about the not significant factors. Here I show you a small selection. That means, for example, we showed that nosemosis is no problem in Austria at all. It is a problem in Southern European countries, for example. It showed also that operation size, which is correlated with professionality, was not a factor in the model. Maybe here we need a higher sample size to see an effect. It also showed that signs of varrosis are not as a good predictor for winter loss. Varroa mite infestation level is a better predictor. This also means that beekeepers really have to actively scan for varroa mites and not only can look for signs of varrosis. And interestingly, it showed also that beekeeper education had no effect in the model. But maybe this is connected with this very easy year with the low losses, and maybe in a year with higher problems, this education could be an factor. In the end, we can advise the beekeepers to treat the honeybees correctly against varroa mite, because it's really the biggest factor in the model. They can also change the queens every 2 years to increase the probability of surviving colonies. When they prepare for wintering, only normally sized and large colonies should be prepared. And they should try to increase professionality and experience. We, researchers and advisors, also have to help. For example, in finding more effective measurements against the varroa mite, to increase the knowledge about predictive markers for colony losses, and also we need to offer high quality training and advice for beekeepers to help them to increase their professionality. In the end, I want to thank the whole surveillance study team. You see, we had bee inspectors, it was a great group of AGA staff. We had help with bee veterinarians in training and interns. We also had the scientific support of the colleagues of the University of Graz, and we really have to thank 200 beekeepers who helped us in the study, and the staff of the bee organizations which helped us with the beekeepers. In the end, we thank the following institutions for the financial support, and I thank you for your attention. Thank you very much, and I await your questions.